Namaste. In the previous video, we studied about pollen pistil interactions and double fertilization. Video, we'll see about post fertilization. Okay, so structures and events. Like we'll see a recap. Okay, after the fertilization, the double fertilization, we know the embryo sac, okay, that has a central cell which became something called the PEC, okay, right. So this is the PEN, right, and we have here is the zygote, okay. So this is present towards the micropylar end, okay. So this whole structure, it's going to develop into endosperm that will be triploid. So it is, okay, triploid in condition. So the endosperm will be triploid, okay. So these PEC and PEN together they form, okay, endosperm. This zygote will develop into embryo. It develops into embryo, okay, right. Now we'll see here endosperm formation precedes, okay, endosperm formation precedes zygote development. So it precedes zygote development, which means that the endosperm formation will begin immediately, whereas the zygote will remain inactive for a short period of time. So it will wait until some amount of endosperm formation takes place, okay. So that is what the zygote starts dividing only after certain amount of endosperm is formed, okay. I told that, okay, after fertilization the zygote remains inactive for a short period of time, right, till some amount of endosperm is formed, okay. Now for what reason? So this is an adaptation, okay, to provide assured nutrition to the developing embryo. So this is to provide, okay, nutrition. The zygote, for the divisions to form into embryo, it requires, okay, a lot of nutrition or energy, okay. For that purpose, right, endosperm has to form first, and then later, okay, after certain amount of endosperm is formed, now the zygote will start using that endosperm and slowly, okay, develops into embryo, okay. Here we'll see, right. So first, as I told, that endosperm formation have to take place, okay. Now the, okay, there are so many types of endosperm formations, and out of that the most common type of endosperm, okay, is nuclear endosperm, okay. So the example for this, okay, type of nuclear type of endosperm formation is Cocos nucifera. Okay, so that is coconut. In coconut, this type of endosperm formation takes place. Okay, let's see. Okay, so here is the primary endosperm nucleus. This is the pen here, and it starts, okay, dividing by mitosis and results in the formation of, okay, many nuclei. So you can see here it results in the formation of, okay, many nuclei, and this side we have is a zygote here. Okay, right. So you can see a lot of, okay, nuclei are formed. So all these, okay, nuclei we call as free nuclear endosperm. So it is free nuclear endosperm. So you see here, this is a free nuclear endosperm and this is a zygote, and this side is the micropylar end. Okay, so once the nuclei finish, I mean, as they go on dividing, okay, on other hand, you may see, okay, cell wall formation starts, okay, taking place. So this cell wall formation, okay, starts forming from outside, and it will be, okay, continued into inside. Okay, and this we call the cellular endosperm, and that is nothing but the white kernel in the coconut. Okay, and we'll see here the, okay, the formation of cell walls takes place from that, okay, outside towards the inside. So here with the arrow mark I'm indicating the formation, okay, of cell walls, okay, from outside towards inside. Okay, so that I call it, okay, cellularization, okay, occurs from outside to towards inside. Okay, so it occurs from outside, okay, to inside here, right? So this is how the endosperm, okay, formation takes place in case of the coconut, and that is the most common type, that what we call a nuclear endosperm, right? Now once this endosperm is formed, okay, of course it will be used by the embryo, right? By the time, okay, this structure develops, of course, into seed, the developing, right, embryo may use up all the endosperm, or it may keep, okay, or leave some amount of endosperm without using it. Okay, so basing on that, we'll see here endosperm, okay, it is completely, right, consumed by the developing embryo, okay, before seed maturation. Okay, so it may, the developing embryo may use completely, okay, the endosperm, right? You will see such condition in case of pea, groundnut, okay, then beans, etc. Okay, and in some cases partially, okay, consumed by the developing embryo, okay, by partially developing embryo, okay, before seed maturation. Okay, so such condition you will see in case of castor and coconut. So such conditions you may see in case of castor and coconut. So okay, so that is okay, right, how endosperm will be utilized by the developing embryo. It may utilize it completely, or it may utilize partially. So in that case, right, the seeds may have some amount of endosperm even after the seed formation. So in such conditions, the seeds will not have any endosperm, right? So here, let's see how this embryo development takes place. Okay, so embryo development occurs at micropylar end of embryo sac. So it occurs at micropylar end, because I told you already that zygote is present towards micropylar end. Right now, the early stages of both monocots and dicots, the early stages of embryonic development will be almost the same in case of monocots and dicots. Okay, so we'll see it as a note. The early stages of embryo development are similar in both monocotyledons and dicotyledons. So it will be same in both monocotyledons and dicotyledons. Okay, so first we'll study about how the embryo development takes place in case of dicot plants, right? And we have to keep in mind that monocot plants are more advanced when compared to dicot plants. In fact, monocots evolved from dicotyledons. Anyway, we'll see first how the development takes place, that is embryonic development takes place in case of dicot plants. Okay, embryo development in dicotyledons. Okay, we'll see embryo development in dicotyledons. Okay, I'm taking here the embryo sac. Okay, so here is the zygote, and here are degenerating synergids, and here are degenerating antipodals. Okay, so this side is the micropylar end, and this side is the chalazal end. Okay, so you can see here these are degenerating antipodals. Okay, and these two are degenerating synergids. Okay, and this is the zygote present, and that is diploid, and here we have is this primary endosperm, right, nucleus. So already we have seen the endosperm formation. I'm not going to discuss about that endosperm formation. We'll see now how the zygote will develop to form embryo here, right. So I'm taking here the zygote. So this zygote divides transversely, so we have to see that so all divisions, in fact, here are mitotic. So all these divisions, that zygote will divide, okay, by mitotic or mitosis only. So it transversely divides and results in the formation of a, okay, a two-celled stage. Okay, so this is a two-celled stage, right? So in this two-celled stage, okay, so one we call it as this is the above one, it is called, okay, the basal cell, right? And this one is the apical cell. So we see there are two cell stage formation, okay, right? So basal cell and okay, apical cell formation we have seen. Now, right, the basal cell, okay, will divide and okay, results in the formation of something called hypophysis and suspensor. It gives rise to, okay, hypophysis and suspensor, whereas apical cell divides and forms a hypocotyle, okay, then shoot apical meristem and okay, also forms something called cotyledons, right? So the basal cell will result in formation of this hypophysis and suspensor, whereas apical cell will divide to form hypocotyle, shoot apical, okay, meristems, okay, right, and cotyledons, right? Okay, so let's say in the next stage, okay, so this is the one, the basal cell that has divided and resulted in the formation of, okay, a suspensor, okay. So the apical cell will form, okay, in this way, right? So you can see here it has an outer, okay, layer of cells, okay, right? So this outer layer of cells is what I call it the dermatogen, okay, and there will be, right, inner layer of cell formation. So you can see here this is a, right, inner layer of cell, okay, right. So this is inner layer of, okay, cells. Now, right, in this case the dermatogen is the one that gives rise to epidermis of embryo, okay. Dermatogen, okay, gives rise to epidermis of embryo. So it will give rise to epidermis of the embryo, okay. And whereas, okay, the inner layer of cells, so these will give rise to, okay, ground meristems, okay, then procambial system of okay, of hypocotyle and cotyledons, okay. So this is what you see here, right? The dermatogen will give rise to epidermis of embryo, and the inner layer of cells will give rise to ground meristem, okay, right, and also to procambial system of this hypocotyle and cotyledons. So this, right, embryo at this stage we call it the globular, okay, embryo, okay, and here this stage we call it the pro embryo, right. So that may be having some 2-celled, okay, 4-celled, 8-celled, and so on and so, and finally it results in the formation of a globe-like structure, okay, on one side, okay. So this is towards the, okay, cavity of that embryo side, and we know this side is the, okay, micropylar end. The same here, the zygote, okay. So this is the location to where, right, this okay, globular embryo has projected into the embryo sac, right. So now the further divisions, okay, will result in the formation of the next stage, that's heart shaped embryo, right. So you see here the suspensor will be still present, okay. Now this the globular embryo slowly changed into an heart shaped, okay, structure. So we can see here, right. So this is a heart shaped embryo, okay. So this is heart shaped embryo, okay. Right. So here I'll mention this is, okay, suspensor, and this portion is the hypophysis, okay, and this is the globular embryo here, okay, right. And in this case the same, you see here, this is the, right, the suspensor, okay, and this is the heart shaped embryo, okay. And then, right, in the next stage, okay, here again the suspensor, we'll see, okay, and this is how the embryo will form, okay, right. So this is the mature embryo, okay, right. So this is still the suspensor, you will see here, okay, right. And so this is the portion where you will find the radical, okay, right. So that consists of the root apical meristems, later it gives rise to the root here, and this is the portion okay, where it consists of a shoot apical meristem. This is called, okay, plumule, and this is the shoot apical, okay, meristem. And here these two are the cotyledons, okay, right. So you will find the mature embryo in this way, okay. So if we see the stages, okay, of this okay, embryo development in dicots, okay, first zygote, okay, and then to pro embryo, then to globular embryo, and then to heart shaped embryo, and finally to a mature embryo, okay, right. So these are the stages of embryo development in case of, okay, dicot plants, right. Now we will see the structure of the dicot embryo, and also after that we will see the structure of, okay, the monocot embryo. Namaste. In this video, we'll say about structure of dicot embryo. Okay. This dicot embryo will consist of two parts. Okay, embryonal axis and presence of two cotyledons. Okay. Of course, due to presence of these two cotyledons, okay, these are all called dicotyledonous. Okay, plants. Right. Fine. So here we find, okay, two cotyledons. So these two are, okay, cotyledons, okay, and this portion, okay, here we have is a plumule which later develop into shoot system. So it will have shoot apical meristems, and okay, here is the portion what we have is a radicle, okay, and that will have the meristems of the root, and that will help in the development of root system. Now if you see here the portion of the embryonal axis, so the portion of the embryonal axis, okay, above the level of cotyledon. So if you, okay, make it, okay, this is an embryonal axis, and you see here these are, okay, the cotyledons, okay. So here when you consider in this way, the portion above this we call it epicotyle, okay, and this is, okay, hypocotyle, right. So the portion of embryonal axis, so this is a, right, embryonal axis. So the portion above the embryonal axis is epicotyle, and portion, okay, of embryonal axis below the cotyledons we call hypocotyle here. So the same will be, right, applied to the structure of monocot embryo also. So in case of monocot embryo, so this is how it looks like, and that is a dicot embryo, okay. But we have studied that the early stages of embryo development, they are similar in both monocots and dicots, but okay, the later stages will completely differ, okay. So here this is the scutellum, and the scutellum is the only cotyledon which is present in case of monocots, okay. Of course the name is the name monocotyledon is due to presence of only one cotyledon, that is, okay, scutellum. So this is the functional, okay, cotyledon here, and we call it a scutellum, okay, right. And so here there are some additional sheaths which you will see in case of monocot embryo, okay, which are not present in case of dicot embryo. Here we have something called, okay, coleoptile. So this is a structure that protects, okay, the shoot apex, and one more, okay, structure called coleorhiza, which will cover this, okay, radicle, and that also has a protective function and helps to protect, okay, the radicle. So this coleorhiza is a undifferentiated sheath. So it's a undifferentiated sheath, okay, and is a protective, okay, covering for root. Okay, so this one is a protective covering for the, okay, root. Now, so if you look at in this case, of course, this epiblast is a the one which we consider a non-functional, okay, cotyledon. So this one is of no use, okay. Out of those two cotyledons, one will become non-functional, only one will, okay, remain functional, okay, and that one we call it scutellum here. Okay, so here this is the shoot apex which will be protected by this, okay, coleoptile, and again an undifferentiated sheath, okay, present covering, okay, the shoot apex. Now in this case, the epicotyle, this epicotyle has, okay, shoot apex. So this one is having okay, shoot apex, and then it also has, okay, something called the leaf primordia. Okay, so this leaf primordia is in fact enclosed in a hollow foliar, okay, structure called coleoptile. So this is what the coleoptile here, it's a hollow foliar, okay, structure, right, and this encloses, okay, something called the leaf primordia. So which means this is the portion which will give rise to the leaves once the germination, okay, takes place, right. So finally we say that in case of monocot embryo, right, it will consist of the monocot embryo, it will be having, okay, just like in dicot, one embryonal axis, okay, and another one cotyledon. It will consist, it consists of one cotyledon. Okay, so that is the reason the name, okay, monocots. Okay, this is the structure of, okay, a monocot embryo. Okay, now let us see the seed. Right. Now the seed is the final product of sexual reproduction. It is the final product of sexual reproduction, and you can say a seed in fact is a fertilized ovule. Okay, next, and these seeds develop inside the fruit. Okay, right. and typically the seed consists of one seed coats. Okay, so there may be only one or two, okay, seed coats. Then again cotyledons, maybe only one or two, and then it has, okay, embryo axis, right? And when we see the seed coats, you will find testa, it's an outer, okay, seed coat, and another one, tegmen, is inner. Okay, so if you see here, so you may find two. Okay, so this is the testa and this is tegmen. So you will find okay, two seed coats, testa and tegmen. The next one, cotyledons. These may be one or two, in fact here, okay, and these are generally, okay, thick and swollen, okay, due to storage of reserve food materials. So they store, okay, reserve food materials, okay, which may be used during the process of, okay, germination. So you may see, like in case of, okay, legumes, that now depending on, okay, presence or absence of endosperm, okay, presence or absence of endosperm, so these seeds are classified into, like, mainly two types. One, I call them albuminous, okay, seeds, or also called endospermic seeds, and another one, X albuminous seeds or non-albuminous seeds. Okay, right. Now in the case of albuminous seeds, they are the one who retain a part of endosperm. So they are going to retain a part of endosperm, and okay, endosperm is not completely consumed up, okay, during embryo development. Okay, is not completely consumed up during embryo development. Okay, you may see in case of wheat, maize, barley, castor, etc. In such, okay, plant seeds, you may find, okay, the albuminous seeds or endospermic seeds, which consists of a portion of, okay, endosperm, right? So complete endosperm will be not used. I think we have already studied, all right, the endosperm is the food or the nutrition for the developing embryo. They may use complete endosperm, or they, okay, right, leave some amount of what I call endosperm. So that portion of endosperm is not utilized by the developing embryo. So here, in case of non-albuminous, okay, or X-albuminous, okay, seeds, here endosperm is completely consumed by okay, completely consumed by the developing, okay, embryo. You will see, okay, such seeds in case of P, okay, and right groundnuts. Okay, so you will see in case of peas and groundnut about these X albuminous or non-albuminous seeds. Now when coming here, in some cases, okay, what we'll see, there will be nucellus still present. Usually nucellus will be used up, okay, nucellus, okay, will be still present even, okay, when the, okay, seed formation takes place. So even at the, okay, seed formation, I mean by the time seed forms, right, the nucellus will be usually used up, but in some cases the nucellus will be still, okay, present. So we call it persistent nucellus. We call it persistent nucellus, okay, and such persistent nucellus we call it perisperm. We call it, okay, perisperm. And okay, such persistent nucellus or the perisperm is seen in case of black pepper. In case of black pepper you may find, and even in case of beet, okay, etc. So these are some red lines in which you will find a persistent nucellus. We call it a perisperm, okay. Now during the formation of seed, okay, so during seed formation, right, so during seed formation the integuments of ovule, okay, so we have seen outer and inner integuments, right, they develop into, okay, protective, right, seed coats. They develop into, okay, protective right, seed coats. And the micropyle, okay, which we have seen when we studied about megasporangium, okay, this micropyle will remain as a small pore, okay, in the seed coat, right. Now this is important. Even the ovule have changed to seed, the micropyle still remains, understood. And for what purpose this micropyle is useful, okay, so this, right, facilitates the entry of, okay, oxygen, okay, water into the, okay, seed during, right, germination, into the seed during, okay, germination. So facilitates entry of oxygen, water into the seed during germination, okay. So that is the function of micropyle, okay, in the seed. Now what happens is, as the seed matures, okay, so as the seed completely comes to a maturation state, okay, the water content, okay, in the seed the water content in the seed is reduced. So very much, in fact, it is reduced, and okay, seeds become okay relatively dry, and it will be only around 10 to 15% moisture by mass. Only that much amount of moisture will be present in the seed, and it looks almost dry, right? And this actually results in the seeds to enter into a state of inactivity, and that state of inactivity we call it right, dormancy. Okay, and that's what the seed dormancy, it's called seed dormancy. Okay, right. So during this process, the general metabolic activity of the embryo slows down. So completely it slows down. Very less metabolic activities will take place. So the seed in fact looks dry completely, but it is not completely dry. Still it has some around 10 to 15% of moisture, and the metabolic activities are almost slowed down. We don't say that they're completely stopped. Right now, now this, when conditions become favorable, if conditions are favorable, that is what is the conditions favorable here? It is adequate amount of moisture, adequate moisture, then oxygen supply, and then suitable temperature. So these are all conditions that are favorable. So if these are met, then seed germination takes place. So seed germination occurs in this case. Now once this we have to keep in mind that ovules will develop into seed, and the ovary will develop into fruit. So this ovules developing into seed and the ovary developing into fruit, these two processes right, will occur simultaneously. So that is proceeds simultaneously. So the process takes place simultaneously, and then the wall of the ovary, the wall of the ovary, right, it's what we call the pericarp, or simply I call it a fruit wall. So the wall of the ovary, what you say, maybe what I call dry, it may become dry, or it may remain fleshy. The wall of the ovary will become pericarp in case of the fruit, right? So when by the time the ovary develops into the fruit wall is what we call pericarp. So that may be having different regions like epicarp, and then mesocarp, and then endocarp, and so right. This pericarp may become dry or it may remain fleshy, and basing on this, okay,